Hi, my name is Bilal, and today I'll be giving you a tour of a lab, showing you important reagents and the overall geography and layout of really important items in the laboratory. Follow me. So here's the lab. I'll be showing you a cabinet right over here. So right over here, we have a lot of different important reagents. Here we have a lot of ionic solutions and buffers, and these are important for constructing buffers with certain pHs or with certain concentrations, and this is really important for certain reactions. Over here we have agarose, which is important for DNA gels to analyze DNA. We have trace metal solutions, which are important for certain reactions. Here we have PBS, which is really important for watching cells in mammalian cell culture. We have EDTA here, which is useful for denaturing certain proteins and for breaking adhesion for cells without destroying cellular surface proteins. Over here we have a variety of beakers and flasks that are autoclaved. And what that means is that they're sterilized so that they're essentially sterile and aren't dirty. Here, you know it's sterilized because this tape on the top has a black mark through it. So that's how you know it's sterilized. This tape is clear when it's not sterilized. Over here, we have a wide variety of items for containing liquids. Here we have some beakers, some flasks and beakers. And there are varying sizes, so for different kinds of solutions and different volumes, you use different receptacles. Here we have different test tubes. These are really commonly used for smaller amounts. So when you're growing a bacterial cell culture, initially, you often put them in this tube. Over here, we have another set of receptacles. Here are graduated cylinders. We often use these for very precise measurements of liquids. So oftentimes when making a buffer, I would use this to get the liquid initially, and then when measuring it to the final volume, I transfer it all over to a graduated cylinder to take it to its final volume.
Hi, my name is Raj. Now that Bilal has finished going over the many types of equipment found in a lab, I'll be going through some of the common chemicals as well as other types of equipment. So if you look over here, we have this chemicals bench, chemical shelf. And whenever you need to make a solution, whether it be for a buffer to run a gel, to make a broth for a particular culture, for any other reasons, you'll most likely be going here to find your chemicals. After you've selected your chemicals, you'll look at a recipe, which you got from your PI or you've found online somewhere to make your particular solution, and you weigh up the exact amount using Weibo, Weibo right here. Afterwards, if you need to dissolve the solid in the liquid further, you can apply some heat, or you can stir it further by putting in a stir bar using this tool right here. If your liquid solution requires a particular pH, you can always use a pH probe to check the pH. So some common chemicals that are often found in labs include agarose, right here. Agarose is often used to make gels, and gels can be used to run, for example, gel electrophoresis. We also have what's called lacto yeast extract, and this is commonly used to provide nutrients in cell cultures if you want to grow different microorganisms. So those are some common chemicals, and now I'll be going over some of the basics of a centrifuge. So oftentimes what a centrifuge is used for is to either A, homogenize a liquid solution, or B, to separate a liquid solution by density, with the heavier solid pellets at the bottom and the supernatant liquid solution at the top. And so I'm going to open up this centrifuge right now, and you're going to notice a couple of things. First of all, you'll notice this lid. And the most important thing when using a centrifuge is to always put the lid back in once you put in the solutions. These solutions will often be contained in what's called Eppendorf tube, or a different type of tube. And you want to make sure that these are balanced. And what that means is, first, both of these tubes must contain the same amount of solution. So this is a little bit more than this, and I'm going to use it in here, but when you actually use a centrifuge, make sure that they are the same amount. So that's one aspect of balancing. The second aspect of balancing is to make sure that if you're using an even number, they're directly across from each other. If you're using an odd number of Eppendorf tubes, you can use what's called a blank that's filled with water of equal volume to counterbalance. Once you're done, be sure to put on the lid close the centrifuge. You can set the speed. You can also set the temperature, which is important for certain reactions, and run the centrifuge. After the centrifuge has started, you'll notice that the speed starts from 0 and builds up to your final speed. A common lab practice is to put your hand on the centrifuge and wait for the speed to build up to make sure that the centrifuge is operating properly. The last thing I'll be showing you today is the freezer. So oftentimes you'll be working with a lot of enzymes that are required to be kept at low temperatures to remain functional. So I'm just going to open up the fridge right now and show you a couple of these. Here we have restriction enzymes. Now these restriction enzymes are used to cut DNA at very specific sequences. If instead of cutting DNA, you want to ligate different parts together, you would use what's called a DNA ligase. Finally, for PCR, you'll use something that's called a polymerase. A common polymerase that's used is TAC that operates at high temperatures. And one of the most important things is if you take something out of the freezer, please be sure to keep it on ice when running your experiment. As a general rule of thumb, if something is in the freezer, keep it on ice when you're using it in a room temperature lab environment.
Hi everyone, I'm Yvette. So now that Raj and Bilal have showed you where the laboratory equipment is and the chemical stockroom is, I'm going to show you a little bit about what you can expect to find at every lab bench. So here we have some common chemicals. This one is called LB broth. It's basically used for bacterial cell cultures, something we'll talk about later in the course when we deal with cloning and transformations and that sort. You'll also have this, which is called 1X running buffer. You usually use this when you're dealing with protein gels and SDS page gels, another component we'll go over later. And then you also have 1X TAE buffer, which you'll use for agarose gels, running it. So this will come up after you do PCRs or after you do restriction enzyme digestions, basically a way that you can visualize your agarose gel that you make. Another chemical we have here is 70% ethanol. So this is a great tool we use usually to disinfect different chemicals in our equipment. It's super quick and easy to use, and it kills most things pretty effectively. We also have different plates here, so I'll show you an example of one. So these are Petri dishes. They're basically just composed of agarose gel inside. So either we have a lab tech do it, or we can do it ourselves really quick to make as well. And it usually just takes about 15 to 20 minutes for it to cool and set. And with that, we can also play all of our different cell cultures and all the processes in cloning. So we'll actually use what we have in here are glass beads. So as Bilal mentioned earlier, we do autoclave our different components just to make sure everything's sterilized and nothing gets contaminated. So these will actually come dry out of the autoclave. We'll help use it to spread our different cell cultures onto the plate. And then afterwards, they'll go into a solution of 70% ethanol once again to clean it before it goes back into the autoclave. We also have a couple other things here. So these are Eppendorf tubes. These are also a different version, different size for them. So normally these will be used to store things like genomic DNA, or you can run some reactions in them. These smaller ones, and there's an even smaller size that we have here, which are super tiny ones, will be used for PCR. And then we also have our burners and our flasks over here. We use them anytime we're dealing with DNA or proteins just to make sure nothing gets contaminated. So when we're dealing with these bottles, we usually have this, and we'll connect it to a glass pipette, so we'll just insert really quickly like this, and that way it's just easier for us to pour out the solution and make sure it's very accurate. So now that I've talked about these different things, I'm actually going to have Howard show you next about our micropipettes, another tool we use whenever we're running reactions.
Hey, so you have something called micropipettes, and every micropipette has a set amount that it dispenses. And it's important to note that every micropipette has a very specific size tip that it utilizes. So, for example, when I use this, one of the biggest micropipettes, I can't fit the tip in here. Please don't try to force the tip into the micropipette or you'll end up breaking the micropipette. Instead, I'll use a bigger tip like this. And then I'll just dispense it into here. Next, we have a vortex machine. So if you want to mix any solutions together, you use a vortex machine. We have an aspirator right here if you want to separate the supernatant. We have gloves, and it's always important to wear gloves in the lab environment. And we also have handy Falcon tubes with solutions that we want at hand. So we have 70% ethanol. We also have DDH2O, isopropanol for isopropanol precipitation, beads for charge separation. And we have a very handy machine. It's called a centrifuge. As you're well aware, we use the centrifuge to concentrate any solids we have in our tubes or anything that we want to have a uniform mixture of. We will use a centrifuge. And I will hand it over to Liv to explain more cool science tools.
Hi, my name is Liv. So over here we have a PCR machine used for the polymerase chain reaction, which we'll learn a lot more about later in the course. Basically, you take those little tiny tubes that Yvette showed you, put them inside the PCR machine here, close it all up, and find the program, press start. What you see here are the different temperatures that the machine runs at in order to basically denature the DNA, which will be this part, and then it has different cycles of renaturing, annealing, and elongation, or annealing and elongation, that you'll basically use to make millions copies of small pieces of DNA, pretty much. After you run PCR, you will bring it over here. You'll run it on a gel. And so Raj mentioned earlier the agarose that you use to run gels. So basically you'll take something that looks like this, and you'll take these little separators and put them in these little notches that we have here, and then you pour the agarose gel that you have made into this little plate, basically. Once you pull these out, after the agarose is set, you'll have little holes in it. You'll come over here and run the gel in this apparatus right here. Basically, these cords, I guess, are running an electric current through this apparatus, which will then move the pieces of DNA based on their size. This apparatus we have right here is used for SDS Page, which is similar, but it's a gel that you basically use to run proteins instead of DNA.